Thank you for that kind introduction. I'm pleased to be here, and I hope I can entertain you in the next 40 minutes about what I think is a coming trend, which is that I think in the next decade or two, we are going to basically sequence every species of living life on the planet. In a poetic moment, I decided to call it the genomic footprints. But we're headed there. And so I thought what I'd do tonight was just basically give you a short history of how we got there and then culminate with the final message, which is this exciting, I think, new trend that's developing. So let's start with the Sanger era. But I thought, since it's a general talk, that I'd very quickly give you a primer on what DNA is. It's a long polymer chain consisting of 4 distinct molecular elements called bases. We can just call them A, C, G, and T, and you can see they're linked together by these yellow molecular components, which are actually phosphate groups. And you can see that the elements are color-coded here to help you. And in effect, that chain encodes the sequence. You can just think of it as encoding the sequence AGA, GT, AGA, GT. You get the idea. Within a cell, it's typically not a single strand, but it's 2 strands where the other strand goes in the opposite direction and has the complementary element where A is the complement of T and G is the complement of C. So both strands contain the same information, and it was this insight that led to the famous conjecture of Watson and Crick about how DNA duplicated itself. In physical terms, I apologize for this weak graphic, but it's actually a double helical structure, and so that's meant to suggest that. In terms of its organization within the cell, it's basically organized into large chromosomes. So this is what's called a karyotype, where each one of these is a chromosome of a fruit fly. And these things are, can be on the order of anywhere from 30 million bases long to 150 million in the case of the human genome. So each chromosome is a single, huge, basically molecular strand. And the topic tonight is how do you actually determine that sequence? Because that sequence is what makes an organism. So for example, you give me the sequence of a fruit fly, you put that into an appropriately conditioned embryonic cell, and you're going to get a fruit fly. It's what encodes for that particular shape and physiology. Okay, so as an informatician, what's the buy-in for me? So the idea is you want to sequence some big strand of DNA. Here is an example of a double-stranded piece of DNA. It's some unknown target. And let's just suppose as an example that it's 150,000 of these elements long. It's a sequence of 150,000 base pairs long. And I won't have time to describe exactly how this is done molecularly, but effectively, in what's called the shotgun technique, basically what you do is you reach in and you grab out a piece of the genome at random. You go in and you grab out another. And for each of those pieces, you basically run a sequencing reaction, which gives you about 750 base pairs. Now, why do you do that? The problem is that the underlying technology for sequencing will not go from left to right for 150,000 base pairs. You can only, the Sanger technique, which I'm about to embark on, will give you about 500 to 800 letters. And that's the maximum. The technology will not give you more. So the question is, if I want to get this 150,000 base pair thing, how am I going to do that? So the shotgun technique is you reach in and you keep sampling, and you do that so that you basically oversample that target. So you want to maybe say oversample it by a factor of 10. So the target's 150,000 base pairs, but you would say, for example, collect 2,000 reads, which if you do the math is 1.5 million base pairs, which is 10 times more data than the actual target. And the idea is that if you've oversampled it, then you should be able to put these pieces together to reconstruct the original. So there are several, so the goal is to build a piece of software, a classic inverse piece of software. The computer science problem is to build what's called an assembler that takes those 2,000 reads, figures out how they should go back together to give you the original target. So it's a classic inverse problem. But there are several problems which I've kind of glossed over. The first one is that you don't know whether the read came from the yellow strand or the green strand. Remember, these strands are directed, so they're in opposite directions. You don't know which of the 2 strands it came from. Another problem is that the technologies are never perfect. there's always some intrinsic error rate of the device. So for example, in the case of the Sanger sequencing, which is what I'm talking about now, there's about a 3% error rate. About, every 3 out of 100 of these letters is wrong. It's going to be the wrong thing. So you have to accommodate error. Another problem is that even though you technically oversampled it by a factor of 10, you've gone in and grabbed 10 times more data than you want, still just by pure random chance, you may not have actually sampled a given particular part of the genome. So there can be what are called gaps. Other problems, which I won't delve into, are that there are artifacts, there are things that can happen in the chemistry that create reads that are not really from the underlying genome. And there's also a problem of repeats, which I'll come back to in a moment, which is that oftentimes there are many segments of the genome that are the same sequence over and over and over again. And we'll talk about how that confounds things. But for right now, basically what an assembler does is it kind of figures out how the reads go together and puts them together into what are called contigs, for contiguous assemblies. Because these contigs contain errors, there's another procedure where you basically look at the concordance between the sequences that all overlap the same place, and you try to fix the errors by basically voting. And then, of course, as I pointed out, just by pure chance, you may not get the whole thing there. You may actually have a hole in your reconstruction. Okay, so the first shotgun project was conducted from, I think, 80 to 82, and it was by Fred Sanger, who won a Nobel Prize for developing, his second Nobel Prize actually, for developing this technique that actually delivers that 750 base pairs that I'm not describing to you, mea culpa. But what I actually read this paper. I'm not sure how many people have read this paper. He sequenced a virus which was 48,052 base pairs long, using exactly the schema that I showed you. And what was amazing in that paper is that basically every idea I think that was pursued in the following 15 years was in that paper. So it was a brilliant paper, short paper. I'm not going to enumerate each of the ideas, but if you haven't read that paper, you should read it, okay? It's really a masterpiece. What I do want to spend a little bit of time talking about is assembly, which was done by his colleague, Roger Stodden, in Cambridge. And basically, at that point, the informatics, the computer science was very simple. Basically, Roger wrote a program that would take two pieces and see if they actually could overlap each other, if they might have shared some relationship. And then he basically started to try to build a tiling, a contig, did it kind of greedily, but there was a lot of human intervention involved. In other words, it was a tool that kind of helped a human being solve this puzzle. And then finally, the sequencing, fixing the sequencing errors was completely done by manual inspection, okay? So what happened basically after that is in 85, some computer scientists got onto this problem, and things improved a little bit. Esko Ukkonen, my colleague from Finland, who's a prolific algorithmicist, was actually the first one to build kind of a complete automated assembler that took the reads and produced an answer. Didn't do a great job, but it was there, and in particular, he also gave the problem structure in that he divided it into the problem of seeing how the pieces would fit together, the overlap phase. Then he had a problem, he had a segment that would, a component that would basically try to find a tiling or an arrangement of the reads and to reproduce the original. And then at the end, he had an algorithm for consensus, which was called, this was called the overlap layout consensus paradigm, OLC paradigm. I'm actually the one who's responsible for the terminology, but Esko was the first one who built one. Ukkonen and Turner then basically looked at kind of, as a combinatorial optimization problem, what's the goal? What are you trying to do with these reads? What's the correct answer? And they said that the correct answer was the shortest sequence that explained all of the reads. In other words, I want the shortest sequence such that every read can be found in that sequence. That turned out not to be correct, but they did a lot of interesting theoretical work about it, and it was the first attempt to formalize the problem. I basically got into this a little bit later, and my PhD student, John Kecioglu, and I basically worked out a model that basically solved this problem of the orientation. We found a graph theoretic model that would basically take care of that. And we basically worked much harder and came up with superior algorithms for each of the components of the OLC. So, but in 1994, probably the most interesting thing happened. So this is kind of a little piece, but it's going to be easy. So, which was called a string graph. And it came up in two separate contexts, which I think is kind of, interesting. There's this consilience and ideas, right? So imagine that this is a genome, is a sequence. So we're so far back from it, right, that I'm just going to draw it as a line. So it's a big, long sequence. It's maybe 1 million base pairs of DNA, okay? And these green segments are meant to be repetitive sequences. So for example, this green segment is a sequence, maybe 2,000 base pairs long, 10,000 base pairs long, that appears not once in the genome, but over and over again. It appears 3 times. And it's essentially the same sequence with respect to the underlying error rate. So, Sanger is 3%. So these sequences are maybe not exactly alike, but with respect to the error rate, they look alike, okay? Then there's the orange, just for example, there's an orange repeat, and there's a red repeat, which occurs 2 times in this genome. So this is just a toy to give you the idea. And the problem, you see, is that with these repeats is that if you're given a shotgun data set and the reads are shorter than the repeats, so the repeats say, 5,000 base pairs long, and the read is 750 base pairs long, so there's that poor little read. If it came from the green piece, right, from one of the green pieces, you don't actually know which one it came from, right? So you can see that it's going to make the puzzle much more difficult, right? Because now you don't know exactly, well, did it come from that one or that one, okay? And so what I'm going to do is basically I'm going to give you a very simple way to think about how to build a string graph. Okay, it has nothing to do with computer science, okay, because I don't have time to explain how to do it in a computer, okay? But imagine the following transformation. Take the yellow line and make it a string, okay, so it's flexible. So I can do whatever I want with it, it's flexible. The yellow is a string. And then imagine the repeats as kind of bars. And what I want you to do is, in your mind, is take all the 3 green pieces and put them together. And take the 2 red pieces and put them together. And take the 2 orange pieces and put them together. And let the yellow stuff just kind of flow, okay, however it wants to. What you get is you get that. Okay, see it? Okay, that's a string graph, okay? You've done it, you've passed the test, okay? Now, it's much more complicated than that, but that's all you really need to see, okay? It's a way, so in some sense, what's interesting about this is the best representation of the information that you have on the reads, okay, absent anything else, right? Because the reads won't tell you how, if you come in from one of these 3 relatively unique contexts, and you come into here, you don't know which way to go out. So you can't correlate the in and the out, but you know that the answer is some kind of walk, right? Some unwinding of these 3 things, okay? So it's kind of I'm just trying to give you an intuition of what this idea was about. And like I said, what was very interesting is I went to a meeting in DIMAX. I was going to present the string graph, and I thought I was really cool. And in the talk right before me, Waterman's student Iduri got up and talked about another string graph on what's called K-mers, which is called the De Bruijn graph. And it turns out that these 2 graphs are kind of the major graphs that are used in trying to solve this assembly problem. But this was a significant advance. And I'll come back to the string graph in a minute when I talk about the Celera story, okay, which I'm about to start on now, okay. So in 95, it was believed, okay, that because of this problem with the repeats, okay, and because of the complexity of the problem, that, if the thing you were trying to sequence was about 150,000 letters long, basically a 2,000, 1,000 piece jigsaw puzzle, that was about as big as you could go. If you tried to do a bigger piece of DNA, it wasn't going to work. You would get confused, you would get lost in the repeats, because genomes are quite repetitive and it wouldn't fly. And so, a very kind of conservative way to do this was to basically think about solving 1,000 piece jigsaw puzzles, right? You can think of these as kind of jigsaw puzzles, right? A bunch of pieces, you want to put them together. And so it was believed that, it's kind of interesting that the 1,000-piece jigsaw puzzle is a puzzle that we do as human beings, right? It was about 1,000, 2,000 pieces. I don't know if there's any real coincidence there. And so the idea was is if you wanted to do something really big, like 150 million letters long, like for example, chromosome, I think chromosome 1 is 150 million long, of human chromosome 1, then the idea was what you would do is you would basically clone big pieces, 150,000 base pair pieces, okay, in an artificial vector. So the biologists figured out a way to take 150,000 piece of an underlying DNA and basically amplify it and keep it alive and purify it, okay? So again, I apologize that I can't spend a lot of time on that. But so what they would do is they would basically shotgun. They would reach in and basically be grabbing 150,000 base pair pieces, so these very big pieces, and then they would clone them, okay, which means that they would be able to propagate them and purify them and keep them going. So they would basically build a shotgun of clones, okay? And they did this with a process called physical mapping because they couldn't sequence them. They did this secondary process, which was quite intricate and somewhat prone to error, I might add. I at one point proved that you actually could not solve this problem optimally. But once you had that physical map that effectively was a kind of super assembly of the bigger target, then you could find a tiling path. You could pick a path that basically covered the underlying 150 million base pairs, and then you could take each of those 150,000 base pair pieces and shotgun sequence them, right? Because we have software for 1,000, 2,000 base pair pieces, okay? There was some, the good aspect of this is that the quality of the outcome was certain, okay, and you were going to get a result. And this was the process that the Human Genome Consortium was using, and in many ways it was appropriate because they were spending taxpayer dollars and they had to make sure that they were going to produce a result, and it was very reliable. But from an efficiency point of view, it was quite difficult because you had two separate processes. In other words, the molecular processes for performing this task and this one were completely different. So you had to have basically two factory floors that had no similarity. And then the other thing was that these clones were never stable. They would change in the test tube. About 4% of them were not the thing that was originally measured. Okay, so you had this underlying error rate in the clones themselves. So that was kind of the state in '95. So, and I started to realize some things. I visited a rather famous ecology geneticist by the name of Fred Blattner in '95, and he had said, well, hey, Gene, what if you just shotgun the whole thing? E. coli was 4.5 million base pairs, right, which was too big a puzzle. And so I thought about it, and there was a statistical theory called the Landerman-Waterman theory, or the Poisson theory around, that was basically just a statistical argument that basically said that, there was a good reason that you would have for not trying to do bigger problems. So for example, if the length of the reads is 500 base pairs and the redundancy or coverage that you do is 6x, then how many contigs do you expect? Or conversely, how many holes do you expect? Regions where you don't have coverage. If you do 150,000 base pair clone, that theory says that you would have 3 holes, 3 places where you got unlucky and didn't have any. If you take the 3 billion base pair human genome, you're going to have 100,000 holes by the theory, right? You're going to have a linear number. You're going to have 20,000 times as many holes because the human genome is 20,000 times bigger, okay? And prima facie, that looks awful, right? Okay. But the theory also says some other interesting things, which it says that the expected amount of the genome that you're going to have actually sampled from is 99.75%, and it doesn't matter if it's a clone or if it's a human genome. You're going to basically have sampled the same amount of it. It's fixed. The length of the contigs, how big are the contiguous pieces going to be before you have a gap? They're going to be 33 kb for the clone and for the human, fixed. How big are the gaps going to be? They're going to be small by the theory. There are 82 base pairs on average, and they're going to be the same for both of them. So another way of thinking about this is if I take 150,000 base pair clone and I shotgun it to, say, 10x, I might get something that looks like that, okay, statistically. But if I take a 150,000 base pair region of the entire human genome, which I've sampled at 10x, I've sampled the human genome at 10x, and I say, hey, this 150,000 base pair, what does it look like? The same, okay? And it's a little, I'm being a little dishonest for my expert colleagues, but I'll fix it in a minute, okay? For those of you that are in the know. So there was, at the same time, there was another development which was becoming very popular, which was the idea of paired-end whole genome shotgun sequencing. So this is a slight variation on the original problem that I gave you, which is that I said you reach in and you grab a piece and you sequence it. Now what you're going to do is you're going to reach in and you're going to grab a piece of a size that you control, say 10,000 base pairs. So you can do that. The biologist, the molecular biologist can go in, grab a piece of a fixed size. And then what was becoming possible molecularly was to basically do the sequencing reaction from both ends. So that you would get a Sanger read that was, so this is 750 letters long here. These are all 750 more or less. But they're all at a known distance and they're in pairs, right? So I know that this one and this one are at a fixed distance from each other, say 10,000, okay? So I can collect them in pairs. So I have a lot more information. I don't have just single reads. I have reads in pairs at a known distance. Does everybody see that? Okay, so now the same problem, same assembler, same problems, but here's the kicker. Now, when I look at this picture, what's going to happen is perhaps this read and this read are paired, okay? So I know they're at some distance, but they're in different contigs. And again, this one and this one, okay, and I could keep going. But you see the point, which is that the fact that these pairs exist tells you that this contig is to the left of this contig, right? And it gives you an idea of how big that gap is, right? Because I know how long these things are. So it orders and orients, so it builds what's called a scaffold, okay, which gives the order and the order of contigs. So you can imagine many contigs, right? And the contigs get ordered by this process, by these links that jump across. So the idea was to come back to this, is that these 100,000 pieces, that looks pretty bad, right? Everybody said, well, hell, I'm going to have 100,000 pieces and I have no idea where they come from. But if I pick the pairs, if I do paired end shotgun, and I pick the pieces long enough, okay, if I design the pair lengths to span most of the gaps and most of the common reads in the genome, then basically I'm going to have all of those 100,000 contigs, or most of them, ordered and oriented, in other words, into a super scaffold structure. So it's not like I have 100,000 pieces and I don't know where to pin them on the map. I suddenly have them in long linear arrangements that I can place on the genome. So basically what I proposed with my colleague Weber in '96 was that we do a 10x shotgun of the entire genome, but that we collect the data in a number of kind of paired ends. So we do a short pair where they're 2 kb apart, a long pair where they're 10 kb apart, and an extra long pair where they're 50 to 100 kb apart. So you can imagine now I have all these links, right, between the contigs, okay? And I'll show you exactly how that works in a minute. And so the idea would then be to scaffold these not into contigs, but into scaffolds where the contigs are linked by virtue of these paired reads, okay? These scaffolds turn out to be very big according to theory, and then the idea was you would just pin them to the genome with some other kind of genetic marker. And the nice thing about this is that it was a single automated process. All you had to do was, Sanger DNA sequencing with the latest automated machines. It involved only a handful of libraries. You basically had to just do 3 library constructions, which was nice. But the problem, the downside, so from a kind of a factory point of view, this was much more automatable and much simpler. The downside was that suddenly assembly just looked bloody hard, okay? Just like, can you do this, okay? And so Weber and I proposed this in '96. This is the only paper in my scientific career where I basically had to go through 4 journals before I would finally get it published, under the condition that it was published with a rebuttal. Okay, I knew something was up. I knew it was exciting at that time. And then something very remarkable happened, which is that in 1998, because nobody wanted to do this, and I couldn't convince anybody to do this, and Weber and I tried. So in 1998, however, Celera was formed to do exactly this. It was as if God had said, here's 300 million, and here's Craig, here's a company, go for it. So and of course, when this company was announced, this is what everybody said, okay? This was from the Times, I think. But it is doable, okay? And the way it was done is as follows. So I'm gonna give you a 3-step process for how we actually did it. So what you do is, remember that graph. So what you do is you wanna build contigs, you wanna put together collections of reads that you know represent unique DNA, okay? Because if you put together a contig over the yellow part of the string, right, not the repeat part, but the yellow part, it's correct. It's going to be because they're unique. So you put them together. The problem is, how do you know it's unique? And you just use simple statistics. You look at how deep the pile is. If you sequenced it to 10x and you build a contig and it's 20 deep or 30 deep, it's a repeat. So we just took all of this, I'm finessing, I'm glossing over a lot of computer science, forgive me. But the trick was to basically find, if you want the geek terminology, was to find maximal uncontested interval subgraphs, okay, that had the right coverage, okay? But we found a technique to basically figure out when we had assembled the yellow parts, okay? So we had the yellow parts. So in terms of this, so here's a genome with repeats in it. Purple repeats, right? What we did is we found all the contigs for the unique bits, the yellow bits, in this picture of white, excuse me, I didn't have time to get all the graphics right. The second thing you do, and it turns out that those are all almost probabilistically, we knew that they were almost all correct. There was very few of them that were likely to be incorrect. Then what we did is we used those pairs, okay? So here are these paired reads, and remember we had them at a variety of distances, and we knew their lengths more or less to an abnormal distribution, and we used those to link together the contigs into large scaffolds. These scaffolds were very large. They were like 10 megabase pairs long, okay? And then basically you can even fill these repeats by basically backfilling. In other words, if I have a read here and I know that it's this far away from its complement read and that places it in this hole, then I know it belongs in that hole, right? So I basically we used to call it throwing pebbles. So we're on the shore and we basically look to see which pairs place reads in the middle of that thing. This is repetitive within the genome, but by itself, it's unique, okay? It's only repetitive somewhere else, okay? But there, the pieces all go together in only one way. So we could actually backfill a huge number of the gaps. And so you get really a quite interesting result. In 2000, this was a 20,000 hour CPU hour compute, which was a big compute for the time. I know it sounds like a small number now, but it was a big number, especially considering that it was a single instance problem, combinatorial problem, right? It was not a weather simulation where you just let time go or make the grid finer. It was a single combinatorial problem with 20,000 CPU hours. So I would say that, basically, so we tied with the public project on the human genome, and these were, I gotta be honest, it was basically not a finished quality genome. There was a lot of work that went on by the public effort after that, but we had at least the first preliminary idea of all of the genes in the human genome. We also did the fruit fly, the mouse, and the mosquito in the same time frame, which kind of indicates how effective the factory was. So the most pleasing thing, I think, about all of this is, while there was a lot of controversy about the results and what happened, is it is now the case that almost every genome is sequenced this way. It's an accepted practice to use a whole genome shotgun sequencing. In fact, it went overboard, and I'll explain that in a minute. And the other thing that I think is important for computer science is that this was a clear case where informatics performed a critical role, right? Because many problems in molecular biology, you can get an 80% solution without being a deep computer science expert. But here was one where people really didn't think it could be done. And so, it was a novel protocol. It was a solution to a daunting problem. So I think that was a win for us geeks. Okay, the short read error. Okay, so what happened after kind of the race to the human genome? What happened basically is that the call was put out that we wanted to do the human genome for $1,000, so we could do medicine, so we could do every human being. And so basically there was a series of technologies, what I call short read technologies, and I'm not, in this talk, I'm not going to explain exactly why they're short and how they're done. But you can see that basically there was a huge increase, right? It went from, basically one of these machines being 1/3 of the 300 machines at Celera to basically in 2018, one of these machines was 36 times the throughput of the 300 machines we had at Celera. So, the quality of the amount of data is outpacing Moore's law. Unfortunately, the reads are very short. I don't have time to describe that, but because of the technologies involved, they would only deliver up to 150 base pairs. And they still today only deliver 150 letters, which is quite short statistically, right, to understand that you've got things together. There were many new algorithmic technologies because of the weight of the code, and a lot of people spent a lot of time on this. But what I want to show you is that de novo shotgun assembly went to hell. Okay, and it was really surprising to me because, for example, at Celera we did melanogaster, and the contig length was roughly, on average, 21 megabase pairs. Shortly after that, with a technology that actually produced more than 150 base pair reads, they got 180 KB contigs. And for two more recent flies, fruit flies, this is 100x of this next generation technology. Contig size is 8,171 base pairs. So I was not a fan. And in fact, I left the field. I was kind of like, well, crap, we're not going to be able to sequence anything with this stuff. Okay, and I knew that in 2003. So I left around 2005. And a lot of useful things have been done with short reads. In particular, a lot of medicine has been done with short reads, and examining the human population has been done with short reads. So, but as a guy who's interested in de novo reconstructions, who wants to go out and sequence the world, this was incredibly disappointing to me. Okay, and as an algorithmicist who's interested in perfecting things, okay, this was also exceedingly bad news to me. So, so the cost became the priority, and it was, unfortunately, there was nothing but short reads. And intrinsically, what they were doing was they were resequencing. They had the human genome. They would take your blood, sequence you for $1,000 in these short reads, and then map all those short reads to that consensus to kind of get a picture of what your genome looks like. Okay, so de novo genomes really just turned, like I said, they went to hell. Okay, and for a lot of things they weren't very good because the gene models of the organism were incomplete. You couldn't understand porology and orthology relationships. Regulatory regions were gone. You didn't have any idea what structural variations might have been in these genomes, and you didn't have any idea of the overall genome architecture. And even, well, I say awful, sorry, I tend to be a bit strong, but even human resequencing was not so good. If you do, so for $1,000, they do a 30x oversampling of your genome. They map those reads, and those reads typically cover 85% of the reference. So they know 85% of your genome. 15% of it's missing. And if you ask over all of the people how much of it is consistently the same, so you're doing a population of the population of the British Isles, about 70% of it is consistently seen every time. Now, again, you can do great things with that information, don't get me wrong, okay? But, from the point of view of really perfecting DNA sequence, I feel that the situation should be better, okay? All right, so let me just go right to the long read era. I've got 10 minutes left? 10, okay, good. I think I might make it. I'm notorious for going over time, just so you know. Okay, so long read sequencing. Okay, so around, actually it started in 2002. I saw the first prototypes of some of these machines, but they didn't really come in until about 2010. And they're very interesting machines. They're single molecule machines, and I'm just going to talk about the one from Pacific Biosciences and mea culpa to Oxford Nanopore, which is also a very interesting technology. But for this particular technology, the idea is you actually take the double-stranded piece of DNA, you run it through a naturally occurring molecule that's actually been heavily engineered called a polymerase, which is responsible for duplicating DNA. And what you do is you watch the reconstruction of the other strand as it incorporates nucleotides, or these elements that have been labeled with fluorescent dyes. So it's literally directly watching the construction of the complementary molecule occurring in real time. So this was a significant feat. And It turns out that you get super long reads. Now you get like reads that are like 20,000 base pairs. So these are long, okay? This is a single read that's 20,000 base pairs. So a lot of repeats are spanned by these reads. But the error rate was really high. The error rate was like 15%, okay? Okay, not 3%, 15%. And initially a lot of people went, this is awful, right? This data, the quality of this data, what can I do with 15% error? But the word random occurs in a very important context, which is that how often it makes a mistake and where it makes a mistake in the sequence that it's reading out is completely random. It's the result of thermodynamic noise, which is not true of any previous technology, okay? And the underlying sampling, in other words, when I reach in and I say, here's a piece, the sampling is pretty much random, again, not perfectly, but close enough from a mathematical perspective that I can assume that it's random. And what I saw immediately as a mathematician, it's not a big thing, any mathematician would have seen it, which is that these conditions, the fact that these technologies are for the first time random with respect to these two aspects, their error and their sampling, meant actually not that 15% was a problem, but that this thing was great, that basically if you do enough sequencing, if you do enough coverage and voting, you're basically going to be able to get a very high quality sequence, and you're going to be able to see everything. You're going to be able to get everything, okay, because you're going to be able to sample the entire genome. So there are going to be very few gaps, okay? So I got very excited about that. So I got so excited in 2013, I started to develop a shop. This is my current repertoire of machines. We did some pilot projects. We sequenced a couple of organisms that are regenerative. For example, the flatworm, you can cut it into 40 pieces. You come back 2 weeks later, you'll have 20, 40 flatworms, okay? The axolotl, you cut off its entire arm, it'll grow it back again, okay? So these are very interesting animals from a point of view of regeneration. The planarium was notoriously hard to sequence. And basically with this new technology, we got 100 times more continuity in the answer, okay, using this new technology over any previous effort people had tried. And with the axolotl, I basically set on it because it's the largest thing I think that's ever been sequenced. It's 32 billion base pairs long. It's 10 times bigger than your genome, okay? And we got those published last year in Nature. So, you know, what I'm basically coming to the opinion of is that we are going to be able to do very high quality, in fact, I think perfect, telomere to telomere. So we're going to go from one end of the chromosome to the other, from the telomere to the telomere. Reconstructions for less than €1,000 is only a matter of time, because like all of these technologies, the price is dropping. I haven't had a chance to talk about price, but price is going down and down and down. These things were very expensive. When I did the axolotl, it cost me like 350 grand to do the axolotl. Today, it would be more like 35, okay? It's gone down by a factor of 10, and it's gonna keep going down. So I think that we're gonna be able to take this technology, and I think at some point, it's gonna get down to the same price point. It's gonna reach that €1,000 barrier. I mean, in the meantime, the short read stuff is down to $300. Okay, but when it gets down to $1,000, then you can imagine going out there and sequencing everything, and we're talking about really good reconstructions. Okay, so I'm participating in some projects, and I'm, you know, as an informatician, we still haven't built an assembler that can do this perfectly. So there's more to be solved. So this was the 15 genomes that were sequenced with this technology, and a number of other technologies that were done at Sanger with my good colleague Richard Durbin, who's here today at Rockefeller with Eric Jarvis and myself, and a couple of these are ours. But you can see that the contig size, on average, is over 1 million base pairs long. And the scaffold, so we used the scaffolding technology, is over 10 million base pairs long. So basically, you know, the chromosomes are in a number of pieces. They're near chromosomal level assembly. On the other hand, the story's still not over, because for some of these genomes, this is the red bar is how repetitive it is, how many of these repeats it is, and this is the black bar is how big were the contigs. You can see that there are still things out there that are repetitive enough that, you know, even with this data, we're still not doing very well. Okay, but for most things, we're doing a great job. I think I'll just go through this one very quickly. The point of the matter is I won't belabor this one because I want to get to the end and let you ask questions. But basically, money's not going to really be the problem. I mean, if I priced doing all 1.5 million known species with this technology, okay, and making some very simple assumptions, it comes out to about 3.8 billion euros. Now remember, the Human Genome Project cost 3 billion. Okay, that was to do 3 gigabase per genome. We're talking about 1.5 million genomes, everything, okay, for 3.8 billion, which in inflation-adjusted amounts is less than the Human Genome Project, okay? But that's not the story, okay? So, but some people might think it's the story. The story is that we can't actually run the machines and get the DNA in the machines fast enough right now. It's good. The problem is throughput. The other problem is that we can't get all of the samples. We just can't access everything. There might be 1.5 million, a list of 1.5 million species that are known, but we can't actually get the living tissue of all of those. So sample acquisition is going to be a huge problem. But I'm really kind of beating on this because there's still a lot of people that are doing a lot of sequencing with the other stuff, and I really feel like that needs to stop because it's like it just means we're going to have to do it again, right? Because these guys are for the record, okay? And we really, but think about it. I mean, I'm totally psyched about it, right? We're going to be able to basically build a catalog. We're going to kind of like be the Linné of our time. We're going to be able to build a catalog of all the life on the planet with basically almost near perfect genomes. And that's what I meant by the genomic footprints, right? We're going to be able to see the footprints of the molecular encoding of life because we're going to see it all. And we're going to see it all at a level that's really going to allow us to begin to understand what's going on. So, there are several approaches. That one is to kind of start one representative of the orders and go down. There are other projects to go deep. There are also projects to look at kind of geographic areas, and these are all important. And so, at one point, I was at the Wellcome, and I made the recommendation that we needed a crash program and automation to handle the throughput problem, and we need to establish sample collection networks, global sample collection networks. So the projects, there are actually a number of projects that are actually underway, so this is not like my fantasy, okay? There are a bunch of crazy people that study bats that want to sequence all 1,400 bats, okay? They're my good friends. There's another group that I enjoy that actually really started this whole thing, whose aim is to sequence all the vertebrates. There are 70,000 different vertebrates. And then there's a group that said, hey, let's sequence everything, the Earth Biogenome Project, okay? But I want to close with the fact that geographic variation is also important. And so in England, you guys should know that you're now engaged in a project called the Darwin Project, or the Tree of Life Project, which is to sequence all of the fauna in England, all of the plants and animals in England, of which there are 60,000 species. In a way, this is a very smart thing because all of the national museums have incredibly good collection, and there's incredibly good knowledge about those 60,000. So actually getting them is realistic. Getting all 1.5 million species, I think we still quite a bit of legwork to do. And then I really kind of want to shout out because I think that this group is actually leading the way. They have a major amount of money from the Wellcome and more to actually proceed, which the other projects do not as yet have. They indeed have a sample collection network with all of the national museums, and they are indeed really going at it to basically get the automation and to get this thing to go. They've got ambitious goals. Let me shout out for that. I think that history is repeating itself. I don't mean to flatter you British folks all too much, but it was the case that the Sanger Center under John Salston, along with Richard and many of his colleagues, was at the forefront of the public effort to sequence the human genome. I mean, they basically developed the techniques and the methods at the front. I mean, at the end, the Americans were the 600-pound gorilla, and they came in and they spent more money. But only at the end, okay? I mean, intellectually, really, Sanger, I think, led the way. And, I think it's repeating itself, actually. So I think it's a very exciting project, and I hope that you'll continue to support it, because I think this is going to be an intellectual leader. So I want to make some final remarks, which is that, I really think that hopefully I've made it clear that I think that this is going to be affordable, and it's going to produce the most beautiful reconstructions we've ever seen, and that it's going to happen. It's just a matter of time. The hard part of being a visionary isn't predicting what's going to happen, it's predicting when it's going to happen. And notice I haven't said when, just eventually. The nascent projects are on the way, and I think that it's going to be, I may be a little bit too old, but for you younger folks out there, I think it's going to be a really exciting time for y'all. Thank you very much.